determining region, number 1, 2, and 3. And number 3, CDR3, complementary determining region number 3, is the most interesting because it kind of captures the most diversity. You can look there, and perhaps this is where the most diversity will be happening. So we're going to be using CDR3 sequence as the proxy for the clonotype. And we're going to say that all the clonotypes from the same individual is the repertoire. So those are the definitions. So we have the problem to study, right? Like we would like to know the distribution of CDR3 sequences over the individuals and perhaps across populations and including diverse population. We want to know what is going on. And one question to ask, like what technologies we can use, like what is available to us to do that. So there is a proper technology to study the immune repertoire called AIR-seq. So sometimes it's called TCR-seq or BCR-seq. That's a proper technology which will look only at TCR or BCR low side and will deliver you the reads, sequencing reads coming from that low side. But the problem with that, especially from the perspective of reuse, we would like to take that existing data generated by the technology and reuse it, is there is not that many samples available, unfortunately, and ancestry is unknown. So we don't know what ancestry or ethnicity those individual belongs to, right? So that's a problem. So if you want to study the immunome, the word perhaps you heard less, so I imagine everybody of you are aware of the genome. That's something become a very common term, even very, very broadly. But the word immunome is not there yet, and the reason is just because we are not doing it. We are not studying that. So you can define one way to define immunome will be the totality, the collection of all the receptor repertoires across all the populations. And we would to study that AIR-seq perhaps is not suitable. We have just too little samples. However, there's another technology, which is RNA sequencing or whole genome sequencing technology, which is used for other things. But we can say, can we repurpose that? Can we repurpose the technology so that we will study that for the population level profiling? And by the way, we are not yet doing population level stuff for immunogenomics because we don't have technology. So if we can unlock, our idea was, if we can unlock the RNA-seq and whole genome sequencing to study the repertoire, then we can do suddenly we can do population level immunology. Okay, so the motivation was that paper which we published a while ago, where we looked at the reads which are discarded. So there is part of sequencing information from sequencing machine given to you, which typically people throw away. And we said, is that right? Is that the right approach? Maybe there is some value in the things people throw away. And indeed, it was some value, and there is many, many things you can do from the reads, what we call unmapped, meaning they were not mapped on the human genome. Things like, like you can do repeat elements, you can do hyper-edited RNAs, non-collinear RNAs, and many, many other things. But the one we're going to be focusing today will be the reads which are coming from VDJ recombinations, the ones which are potentially coming from CDR3 sequences. So those are the numbers. If you take a typical experiments, we're only using 82% of the data. So I think it's a generally important point that we should be using. We should try to use all the data, and we should be always very careful when we throw away the data. So in this case, you can see that we have 0.02% of reads what we are interested in, right? Like the reads which are coming from TCR and BCR. So yet we hope that sufficient number which will allow us to study the repertoire. Okay, so the way it works, so we have RNA-seq molecules, and then which are sequenced. So what you're going to do, you're going to shred your DNA into the fragment, and every fragment will be sequenced, what we call the read, so kind of read out of the DNA, in this case, RNA. So, and then the genome, so it's available to you. So what you can do, you can take your genome and match the reads, which is coming from the sequencing machine, and then the genes, the coordinates of the genes. So what you can do, in this case, we have two genes, green and orange. So in this case, you see that there are more reads coming from the green than the orange. So you can say that the expression of the green gene estimated as number of reads matching that gene is higher. And this is what we do when we do gene counts. And having that information, you can look at the differentially expressed genes across conditions and diseases. You can study the RNA-seq structure, RNA structure. You can assemble the novel transcripts. But what is common between that? You're never looking at the reads which are in gray, which are unmapped reads, and you will literally throw them in the trash bin. So we would like to use those reads to study the adaptive immunity. So what that means in terms of coming back to that picture, what that means in terms of the reads. So if the reads are entirely mapped to the V gene, those reads will be mapped, meaning because the V gene is actually part of your part of the many V genes are part of the reference genome. But if the read is spanning the recombination, it's coming kind of part of it coming from the V, part of non-template insertion, and so on, that is hard. So there is no currently tools, bioinformatics tools are capable of taking that, like capturing it. So this is exactly what we did. We did the tool called IMRAP, Immune Repertoire Profiling tool, which allows you to utilize those kind of reads which are otherwise hard to capture. So the method is it's quite, no, let's not say extremely, but it's quite simple. And I would like to walk you through just to give you idea, like what is behind those bioinformatics tools when you run them, because there is a certain algorithm behind, and this is actually quite simple one. So it's a good opportunity to kind of appreciate and dive into that. So the way it works, so we are seeing a read, and the read is nucleotide. So we're going to convert that to the amino acids to all the frames, and we will hope that read will have CDR3 sequence inside. And what we do, we use the definition of the CDR3 sequence right away. So the definition tells you that that CDR3 sequence always starts at the cysteine and ends with the phenylalanine. So if that's the case, what we can do, we can scan the read from the left to the right until we find the cysteine, and we can do the same from the right to the left until we find the phenylalanine. And that's our candidate CDR3 sequence. So what was not that hard. Now, next, we need to look at the flanking region, and we need to match them with the V genes, like this, right? So we take that piece, which is not highlighted in yellow, and then we try to match it with V gene. Is it a match? Not yet, right? V1 is not a match. We try V2, and we try V3, and that's a match. Great, and then we're gonna repeat the same procedure for the J gene, or for the flanking region on the right. And that's no longer candidate CDR3 sequence. That's true CDR3 sequence we're going to report. There are a few details here, which I will briefly say. So sometimes it's a short. It depends on the technology, but some technologies are delivering 75 base pairs or 50 base pairs historically. So if they're so short, can we still use it? Because if they're short, what can happen, so what can happen, they can only overlap V gene, but they will be too short to overlap the J. And the question, can we use that? And we thought, okay, so again, that kind of idea not to throw the information away, we decided to use them. So the way we use them, so we keep all the reads which has this partial CDR3 sequence, like this, right? We have all of them on the left. And we're going to have the reads which overlaps only the J on the right. And then we can look at the overlap of those CDR3 sequences using the structure which is known as a suffix tree in computer science, which allows you to do it very, very quickly. So an example, right? So this is the read overlapping V3, but did not overlap the J, was too short. And this is the read overlapping J, but does not overlap the V. So if we look at the overlap of 5, and empirically we demonstrated that 5 was not enough overlap, then if there is that overlap, you can merge it, and suddenly you will get a full CDR3 sequence. We are aware of the errors. There are errors in the data, so sequencing technology is not perfect. So there are errors, what we call sequencing errors in the data. So if you have two CDR3 sequences which are too close to each other, there is a chance they will have some error. right? So we will collapse that. So we'll do some clustering there. Okay, so that was the algorithm. Now what we do, and we'll try to briefly kind of go through that to be on time. So what we do in bioinformatics, as we develop the algorithm, we need to know how it performs. Like, is it accurate? Because we don't know. And we need to compare to others and hopefully be better. Because that's a justification why people need to care about what you did, because it does better under certain conditions. So this is what we did. We simulated the data, meaning we assume we know how the sequencing machine works, and we simulated that on the computer, and we look at the results. So as you can see that our method was quite nice across different sequencing parameters and better than others. But that's just the start, because simulation data is always oversimplification of reality. So we found real data. So for real data, we took BCR sequencing as the truth, and we compare to that. So what you can see on the plot, so y-axis would be the truth, or the gold standard, better say, and IMRAP, us, will be on the x-axis. So what you can see in the majority of the cases, we are doing quite well estimating the relative abundance of those clonotypes we have assembled. We also asked the question, what is the fraction of the repertoire we are able to capture? And you can see that we are doing quite well. On average, we are capturing around 50% of the repertoire, especially of the clonotype which are higher frequency. In other words, if the clonotype is rare, it's very, very hard to capture it. Okay, so I think to be on time, I might skip this part and we'll just briefly mention, and then we'll go to the conclusion. So it's always, I think, there's a lot of, how should I say, curiosity and rigor you need to have when you're developing the tool. So that was just the start, right? Like, we spent the time, we developed the tool, we validated that. But there are always other questions to be asked. So in this case, we say, does what we do have any application in the clinical setting? Like, can we make a case? And this is what we have tried here. We said, will the tissue type and the repertoire type play a role in the accuracy, right? So, and then under what condition our results of our tool can be used in some sort of clinical settings? Because oftentimes when there is some cancer cohort or other cohort, people already done some sequencing technology, meaning perhaps they done RNA-seq, they done whole genome sequencing. And if we can come and say, hey, for those samples, we give you really, really accurate results, and you can use that in that study, I guess it will be appreciated. But we need to know those conditions, like what tissue types, what repertoire types we can use. And this is what we call benchmarking. And this is, I think, as important as the development. So basically, you try to make a designated effort to study the performance of the tool. So the tool was already developed, but you want to study it very, very carefully, under which conditions the tool will perform well. So okay, so I think we'll skip some slides and we'll go to the conclusion just to be on time. Let me see what